Okay, so for then our neurotransmitters, right? Sure that I'm recording. Perfect. Um, Now we're going to talk about what is going on the postsynaptic cell. What is receiving that chemical release in order to allow for the signal to continue onward, right? This is important. We need to know, again, do I make it presynaptically as a neurotransmitter? Do I have a receptor on the postsynaptic cell that can receive it? So again, receptors are just a folding of protein, an RNA protein, that are on the plasma membrane of the postsynaptic cell. Again, so where am I? A little whiteboard, move the last here. All right, so my presynaptic cell, we've just covered how I can release my little vesicles, right? And so this is exactly what I'm talking about now. I'm focusing on these little blue dots here. These are the receptors that are going to receive the neurotransmitter, right, from one way to the other across my pre to post. So I must have receptors on the postsynaptic cell that can receive. And we have two major types that we're going to look at. First are ionotropic. This is a term we should feel pretty comfortable with from cell biology. These are fast acting, and they are exactly as the name implies. They're moving some version of an ion, right? They are ligand gated. We're going to look at this in comparison. Versus our metabotropic, again, terminology from cell biology. Metabotropic being slower. They utilize what we call a G protein coupled receptor. So for my ligand or in my ionotropic, right, I am going to have a generalized structure that I'm moving, up for, because again, I have to get over the hurdle of my hydrophobic and hydrophilic aspects of my membrane, right? The biggest hurdle being my hydrophobic pores. So now I've got a receptor similar to what a channel does, but in this essence, we're calling it a receptor because something has to bind in order for it to open. Now it's not like our leak channel that just sort of willingly lets potassium in and out, right? Now I need to bind something to the receptor. I need to put in my key to open the lock. I need to have my ligand open the lock. So here, the ligand is the neurotransmitter. The key is the neurotransmitter. This is why you've just released it from the presynaptic cell. That key opens up the lock, and now what do we see? Boom, my ion can flow in. What are we going to probably guess that ion to be? For the most part, we're looking at things like, again, sodium. We will talk about some other mechanisms, things that can also hyperpolarize the cell, things like fluoride. Fluoride is a negative ion, right? For the most part, we're talking about changes in membrane because we need to push the signal on in the next cell. This is different in comparison then to our metabotropic. Notice here, there's a lot of steps that are going on. More steps, more numbers equals time. Time means slow. So for my metabotropic, what would I want you to do? What would I want you to know? Again, I still need my ligand. There is my, right? Maybe that's something we should explain the difference of. If I am a channel, right? I don't need a ligand to open. I just need whatever it is that readily flows through me to come through, right? Some are made for water, some are made just for potassium, some are made for sodium, some are made for other things, right? I don't need a ligand. I don't need a key. For a receptor, though, I need my key. My key is my ligand, right? Again, I can see that right here. Here's my little neurotransmitter. Here's my little key into my lock. And now the process can begin. So again, in our metabotropic, it's slow, takes lots of time. I first need to bind, then I have to activate my G protein receptor. My G protein receptor goes off and does other things, right? And eventually what it could do is it can activate these secondary messaging pathways. Secondary messaging pathways may lead to things like ramping up protein production. I remember when I said if I'm going through different versions of tolerance, I'm going to eventually lead to restructuring the number of receptors that are in my postsynaptic cell. Maybe in the front side, as I'm becoming addicted, I'm upregulating a bunch of receptors because I've got more neurotransmitter being, oh my gosh, I got to catch all these keys, right? But then over time, the system says, those keys coming at us like that, that's not good for us. That's not very healthy. Let's start down regulating some of those receptors. All of that would be done, right? Because you have to go to the DNA, make more RNA, make more protein. That takes time. That is through metabotropic. And so really what they can do is two major things. They can open an ion channel, which again can lead to activation or inactivation, or they can stimulate or inhibit our secondary messaging through these enzymes. And enzymes and these secondary messengers, again, can either ramp up a signal, make a signal very loud inside of the cell, or it can activate these secondary messaging pathways called protein kinases. This is as far, this is as far down this rabbit hole as I would like to go. Maybe I ask you, fill in the steps. Maybe I ask you, what is the difference between metabotropic and ionotropic, right? This is slow, this is fast. True, false. Metabotropic receptors need a ligand, true. Channels need a ligand, false, right? How do metabotropic, give me two ways in which metabotropic receptors work. They can lead to ion channels opening or closing. They can stimulate or inactivate effector enzymes. What can second messaging systems do? They can amplify signals or they can turn on protein kinases. Again, very simple high level here. So this all leads to this concept then of where we're going for the remainder of the class. We have to lay down the foundation of what the nervous system does. First, we have to tell us, what is pharmacology? What is neuropsychopharmacology? How do we even define that? What does that look like and why? Why should we care? How do we then define, okay, this neuro part of it? I guess I have to understand what neuro means. I have to understand how cells communicate inside of that neuro system, right? Because then I can understand, how do drugs mess it up? That's always what we're doing in science. How does this mess it up? So as far as synaptic transmission is, as far as we are understanding, action potential, electrical to chemical across from pre to post, there are lots of different places, tons of different places that drugs can inhibit or enhance this process. So drugs can affect the overall rate of neurotransmitter synthesis, how much is being made, how little is being made. How often are we releasing? What is being activated on the postsynaptic cell? How are receptors binding? And again, the last, that we've already kind of looked at, how do I clear it? How do I make sure the bathtub is ready to be used? So these are just examples. You don't have to know these in particular, but this is just some different versions of drugs that we may have heard and some that we're for sure going to talk about in this class. And you can see where their effects are, right? Haloperidol, right? A very powerful analgesic. This is something that is binding to dopamine receptors. Nicotine is going to activate these ACH receptors. LSD is going to have an effect on serotonin receptors, right? And so again, this is all just simply showing you that once we start playing with drugs, we have to understand how does the system work first without the drug to be able to say where is it messing up momentarily. You activating or inhibiting, right? In the case of lithium, you see a big circle with the, it's shutting down the potentiation. It's shutting down that signal. All I want you to get away from this, right? Maybe I say, out of the 11 different types, the 11 different examples that we're given, give me 2 to 3 different ways, 2 to 3 different mechanisms in which drugs can alter synaptic transmission. You probably wouldn't even need to go to here. You could just use this slide. It can affect the rate of release. It can affect the synthesis, right? So all of this to say that as we get towards addictive states, eventually the environment, the frequency, the time, the amount, background, fundamental, formidable aspects of how your cognition was formed over your lifetime, right? That learning and memory leads to things we like to call plasticity. Plasticity is a beautiful, beautiful thing inside of the brain. It is this concept, but again, the old dog new trick situation. Plasticity is what affirms the new trick situation. Your brain maybe is not constantly regenerating neurons, but it's constantly modifying the synaptic dendritic. So all those little dendritic spines that we see, those constantly modifying and changing shape, right? Because you're being exposed to new experiences, new environments, new things, new drugs, right? Maybe something happens where you need to take a drug now. All modification of synaptic plasticity. So there's 2 major types. We can have functional changes, which again is synapse strengthening, or we can have structural changes in which we can see the growth or loss of dendritic spine. So let's see. In structural neuroplasticity, right? It's the ability of the brain to physically change the structure due to learning and memory and experience. I'm changing the overall, structural. I'm growing, I'm feeding, I'm modifying, I'm physically doing the physical change versus the functional. Again, I'm remodeling how my system is integrated based upon, again, maybe an injury, exposure to drugs. So again, this is just another way we can look at it. I can have a series of different, right, synapses, pre to post, that something happens. In this case, we'll just use the example of drug use and abuse. Is that something modifies and takes out neurons, right? So now I only have 3 of these synapses, 3 of these neurons in communication in this overall, model. But what happens? The brain is very clever. It's going to say, okay, well, I've lost that, but that was an important part of my communication, part of my plasticity as part of my learning and memory. Now, during here, learning and memory may be compensated. But the beautiful part is that given time, I'm not going to regenerate a new cell, but I'm going to regenerate these dendritic spines that are going to hopefully help compensate BD again so that D starts firing and the system continues, right? So Here's an example of a drug. These are pictomicrographs of the dendritic spine density in the nucleus accumbens. An area we're going to learn a lot about when it comes to addiction, especially with dopamine. And these are rats that have been exposed to control, saline, or they have been repeatedly exposed to MDMA, ecstasy, Molly. What we see is that those that were exposed, there is an increase in synaptic density within this area. And this is thought to be due to the rewarding properties of this drug as part of the abuse. Now, again, just because we have more doesn't mean better. We'll come back to that. The last things of this particular chapter just covers one of the last major aspects of system that we need to think about, which is cell A talks to cell B. That's very important. But we also can see that hormones act as neurotransmitters, but they are taking a very different route. Again, this is going to take time. So this is via the endocrine system. The endocrine system uses hormones, and hormones are secreted by an endocrine gland. I've got a picture coming in just a second. And that information, that hormone then travels in the bloodstream to get to its downstream target, right? I still must have the same concept. If I'm making a neurotransmitter and it's being released, it must be received somewhere. So my target cell must have a specific receptor for my cues to lock into. What we see is that many times a lot of these substances can act as both a neurotransmitter and a hormone. So there are things that can do double dose. They may affect the brain, but they also may affect the endocrine system. We're going to talk about that too. So this is just an example of just a broad overview of the endocrine glands inside of your body. And you're probably very familiar with these, right? Especially with your adrenal cortex and your adrenal medulla. These are very important for your flight or fight response. Again, am I going to run? Am I going to stay? Versus absolutely, we know that hormones are extremely important in reproduction. They're also extremely important in puberty and maturation, right? In both of the sexes. When we say gender, sex, male. So hormones can have two major mechanisms of action. They can be a peptide. What was that term? Protein. Takes time. Hormone. If I'm using time to make the thing, it probably is going to activate those that are metabotropic slowly. So this is things like insulin. We'll talk about all of this to come. Insulin is something your body naturally makes by hormone. It helps to regulate your blood sugar. I can also see that I have steroid hormones, and these steroid hormones act as intracellular receptors for the nucleus, which turn on gene transcription. Boy, that makes sense. Right? I have to upregulate. I have to be responsive to long-term action. So again, in our adrenal cortex, glucocorticoids, or steroid hormones, these are given in response to stress. Now, your immediates are your fight or flight, but these glucocorticoids, when these start to turn on, you are turning into long-term stressors. This is when we start to see people, they start to puff up a little bit, or they get extra weight. They turn on autoimmune disorders because you're now bypassing and you're turning on gene transcription that's modifying. Now we're going back down to that DNA, RNA protein, right? So to be continued. All right, the last thing, why is this important? I'm sure you can figure out hormones play an important role. right? They still are acting as neurotransmitters in a lot of different aspects. And again, there are things that we have to look at as far as the substances that can be affecting the endocrine and acting as neurotransmitters. So drugs can alter the endocrine function, things like BPA, things that we hear about in plastics, BPA free, BPA free. What does that mean? It is an endocrine disruptor. It can bind to and disrupt. And we've shown this multiple times in humans, rodents, things like zebrafish. It disrupts in a lot of different models. It is an endocrine disruptor. It can lead to things like premature delivery. It can affect sperm quality. It can lead to fertility issues overall. The implantation of the embryo into the uterus, right? Cancers. Again, hormones can modify how drugs are creating the behavioral response. We're going to definitely look at this between alcohol and the differences between sexes. And again, the neurotransmitters control the overall release of the pituitary gland or, systemic release out. The brain releases neurotransmitter from the hypothalamus to the thalamus to the pituitary. And the pituitary sends out its own version of neurotransmitters that then downregulate or upregulate all of these downstream receptors in these target organs of the endocrine. So that is it. That is everything that you will need to know for chapter 3. I will put together a guide document for you, but for right now, you've got all the information that you need to be able to finish this chapter. You can get on it and begin studying ASAP because it's a lot of information. So I will see you guys on Monday.